Hi everyone, my name is Mateo Avila and I am excited to present our work on a similarity metric for neural network models in evolutionary neural architecture search. This research was conducted by myself, by René Groh and Andreas Kist. So here is the main outline. I will briefly discuss the background of evolutionary NAS and sequence alignment. I will explain how we compute the similarity score between neural networks in our search space and how this score measures the population diversity in evolutionary NAS. Then we propose an algorithm to incorporate our similarity metric into the NAS pipeline in order to optimize both exploration and exploitation phases. And finally, in the last section, we will present the use cases of our similarity metric, including tests on three popular datasets, as well as the limitations of our search space. So, evolutionary NAS is an approach that, inspired by biological evolution, searches for the best neural network configuration between a population. So this process uses genetic operations to sample new populations, as shown in the general pipeline. And one of its advantages is that it leads to the discovery of novel and efficient neural network architectures that might not be found using other search methods. And it does not require gradient information. Its disadvantage is that it can be slow and often requires training and large number of models. Sequence alignment in biology is a method that is used in bioinformatics to arrange sequences of DNA or proteins to identify different regions of similarity that may indicate functional or evolutionary relationships. For example, in this chart, the 53 proteins are crucial tumor suppressors that can help maintain genomic stability. So aligning these sequences reveals important conserved regions are critical for their function and highlights evolutionary relationships across different species. So it means that similar species typically present more similar alignments scores and sequences. So now let's see how we adapted this sequence alignment concept to estimate similarity between model architectures. So the question we aim to address is how can we compare model architectures within our generations and monitor the evolution of diversity in our evolutionary NAS. So first, we need to understand the structure of our search space. We use a chain-based search space that is composed of three main groups of layers: feature blocks, that is in red, a global pooling layer, and a dense block that is in green. For features block, we incorporated primitive layers, mainly like convolutions and average pooling, as well as basic blocks and bottleneck blocks from ResNets. And to compute the similarity metric, the first step is to represent this architecture as a sequence. So we assign a unique character to each type of layer, and since each layer has multiple parameters, we store these parameters in a dictionary. As a result, the architecture sequence is represented as an ordered dictionary that captures both the types and parameters of each layer. The second step is to select the two models represented as sequences to compare and compute the alignment score based on the matches, mismatches, and gaps created to align the sequences. How we compute this alignment score? So it's performed using BioPython library, which implements the Needleman-Wunsch algorithm that is based on dynamic programming. So by default, this sequence alignment function only considers matching and mismatching based on the characters. So we modify these functions to also take into account the parameters of each layer, like the kernel size and the padding, etc., making the matching and mismatching process dependent on both layer type and parameters. So the resulting alignment score is not normalized and depends on the length of the sequences. Therefore, we normalize the scores to get a relative similarity metric that allows us to compare any pair of architectures in our search space. And a score of 0 indicates that the models are completely different, while a score of 1 means that the models are exactly the same or have the similar architectures. So we can create a similarity matrix with the pairwise similarities between all individuals of a population, where the diagonal represents the self-alignments. So to measure the diversity of a population, what we did was to calculate the mean of diagonal similarities. So a low mean similarity indicates that there is a high diversity in the population, while a high mean similarity suggests that there is a low diversity in the population, as shown in the more yellow heat map. Now let's see how we can use this similarity metric to guide the diversity during the NAS process. So by tracking the mean of the population similarity in a run of CIFAR 100, we observed that our NAS could have converged faster if we had accelerated exploitation rather than maintaining like a high diversity during the many generations. So the question is, can we use our population similarity metric to optimize the exploration and exploitation phases? So we modify our pipeline to include a dynamic genetic operations that are tuned by the diversity score in each generation, as shown in the pipeline. And this approach, what it does is to generate new populations until the median similarity difference between consecutive generations falls within the limit set by our control function. So this control function determines when to allow diversity to increase and when to reduce diversity. And for testing, we propose a rigid control function that limits diversity increase after generation 10, try to accelerate convergence in NAS. And now we will present some use cases we identified for the similarity metric and the results of the diversity guidance strategy. So we conducted two sets of experiments, one naive without diversity control and one with diversity control. And we ran each one with different seeds on ImageNet 16, 120, CIFAR 10, and CIFAR 100, and user pair parameters and data augmentation as in NAS Bench 201. So the primary use case of our similarity metric is to monitor the diversity of the NAS process. So it allows us to identify potential issues in the selection of genetic operations or during the selection step. For example, in one of our run of CIFAR 100, the mean similarity score was very high in generation 5, as shown in the figure, which could indicate a premature convergence of the NAS, which is not desirable. And a second case we find interesting is representing the similarity matrix as clustergrams that are combinations of heat maps and dendrograms, and is commonly used in gene expression analysis. So by using these clustergrams, we can identify cluster of very similar models. And for example, again in generation 5, we observe a large cluster of very similar models, indicated also undesirable low diversity in the early generations. Third use case, when using the sequence representation with alignment charts, is to visualize the evolution in the best found architectures per generation to understand which layers were mutated and which ones were preserved. We can also use the similarity matrix to identify outliers or inconsistencies. For example, if there are very similar individuals with significant differences in fitness, this could indicate design or training errors, or it might highlight important genes that are crucial for evaluating the task. We also present the best models found for each seed on ImageNet 16 in this case in an alignment chart, and all of them are very different in their architectures and in their sequences. So this highlights that there is a dependency on the initial population and population size for the effective exploration of the search space. And we also group and identify the common blocks in the top performing models, observing that the most frequent pattern, as shown in the frequency distribution of the individual blocks, is the bottleneck block, is represented by a T, plus a device convolution that is represented by a D. So, and the device convolution plus a batch normalization is the second most common block. Regarding the diversity guidance algorithm tests that are shown in red, the results indicate that NAS follow our control function, which focus on preventing an increase and oscillation in the diversity after epoch 10 to improve convergence. So for all the three datasets, the median highest fitness was higher when using the diversity control compared to the naive approach. But this difference is small and highly dependent on the initial population, so it's necessary to test new control functions at larger population sizes in the future. So regarding the limitations, this work primarily focused on the design and use cases of the similarity metric, and future work should aim to test this approach with larger population sizes and explore different control functions, as I said, also different search spaces. And currently it is designed to work just with chain-based search spaces. So future work could also involve adapting it for cell-based search spaces, which are more common in NAS. Thank you all for your attention.